can see my screen, right? Yeah. In the last class, we just came to one small discussion where what is a genetic material, and there are two types of nucleic acids I told. That is deoxyribonucleic acid, and then you have ribonucleic acid, which is called as a DNA and an RNA. And in deoxyribonucleic acid, which stands for a DNA, correct? No. So we talked about some of the small properties, like very very basic properties of what is DNA. Today we are going to discuss how what is the composition of a DNA and how DNA is structured. Okay, so if I look at the composition, how the DNA is made up of, I know that you have studied this even in your 12th standard textbooks, but it is quite important for us. There are some aspects that you will also think that okay, this guy is telling something new, so you just concentrate. Okay, I'll not specify on something that is very much in your textbook, so we'll just brush up on it. So let's go here. So I told you DNA is composed of three key ingredients. That is, one is the deoxyribose sugar, and then we have the phosphate group, and then we have the nitrogenous base. Okay, so three ingredients are required. Like that yesterday I told you that like to make a coffee, you require like the milk, and you require coffee powder, and then you require the sugar. Sometimes sugar. Or let's say the ideal coffee. So these three. Similarly, to make one DNA molecule, these three important ingredients are very, very important. That is a deoxyribose sugar, a phosphate group, and a nitrogenous base. Got it? No? First, let's see what is deoxyribose sugar. So when you look at deoxyribose sugar, what type of sugar it is? It is a pentose sugar. So what is the meaning of a pentose sugar? If I ask you to write a formula for a normal glucose molecule, which is our sugar, what do you tell? You write it as C6H12O6. So if you look at the number of carbon atoms here, it is 6. So it is a hexose. Okay, there is 6 carbon atoms. So this is a hexose sugar. So if there is 5 carbon atoms, we call it as a pentose sugar. So this particular sugar, deoxyribose, is made up of 5 carbon atoms, okay, which are designated as C1, C2, C3, C4, and then you have C5, like this. So they are arranged like this, okay. Now you have the formula for this pentose sugar is C5H10O4. So C5H10O4 is the formula for your deoxyribose sugar. Got it? So then you have is a phosphate group. The phosphate group is usually a phosphoric acid. Okay. The phosphoric acid's formula is H3PO4. Correct? No? H3PO4. So this is a molecule which acts as a donor group of a phosphate group. So, sir, is H3PO4 completely present in the DNA molecule? No. The phosphate group of the phosphate group of the DNA molecule comes through the phosphoric acid. You get my point? So the phosphoric acid comes and forms the phosphate group, but you cannot see the whole phosphoric acid. That's common sense, right? So the phosphate group comes from the phosphoric acid. Correct, right? So the next one is you have, uh, you have is the nitrogenous base. So this is an important term. A nitrogenous base is containing compounds that is mainly divided into purines and pyrimidines. So what are these nitrogenous bases? So these nucleotide bases or nitrogenous bases, okay, are actually heterocyclic structures. So now you can divide this into two types. You have adenine, guanine, cytosine, thymine, uracil. So these are the five purines and pyrimidines combined, or nitrogenous bases we have. So we divide these whole nitrogenous bases into two important categories. One is purines and pyrimidines. On what basis do we divide these? Why do we have to divide this entire new nitrogenous bases into two categories? What is the base of dividing them? So the answer for this question is very, very simple. So the structure of these molecules and the composition of this molecule is completely different. So that is why we actually divide them into two identifiable groups. For example, if I take purines, okay, the purines, if I look at them, they are heterocyclic in nature. So what is a heterocyclic? Heterocyclic means if you look at this structure, it has 6 ringed structure. This is a 5 ring structure, 5 atom ring structure and 6 atom ring structure. Here also you can see that this is a 6 atom ring structure. This is a 5 atom ring structure. See, what you have to observe is the examples for purine is adenine and guanine. Okay. Now, if I look at this structure, the 6 ringed structure, it is called as pyramidal ring. Okay, it is called as what? Pyramidal. Okay, pyramidal ring. So this is called as a pyramidal ring. Whereas if you look at what is this five ring, so you have another ring which is of only five atoms. Correct me? There are five. Here it is six. So this ring is called as imidazole. Imidazole ring. See, when you talk about the purine, a purine is a combination of two rings. One is a pyrimidine ring, other one is an imidazole ring. See here, this is a pyrimidine ring and this is a imidazole ring. So this both will combine, okay, to form one compound. So that is your purines. So what are the two types of purines you have? You have adenine and then you have guanine as your purines. So what are purines? They are heterocyclic compounds. So you should tell that it is hetero. Hetero means different, right? So two different types of cycles are there. One is six atom, other one is five atom. So what is the composition of these rings? If you look at a pyramid ring, okay, you can see that it is made up of four. Sorry, excuse me. Yeah, it is made up of. Oh, sorry, it is made up of four nitrogen and two hydrogen or two carbon, I think. Yeah, four nitrogen and two carbon. Here, if you see the imidazole ring is made up of three nitrogen and two carbon. So this is the composition of this ring. You know that, right? Whenever the number of atoms or the structure of molecules vary, they definitely have a completely different function. For example, if you see the difference between H2O and H2O2, you can understand the properties of the compounds. Similarly, even a small changes in the atoms matter a lot. So now you understand, right? Why do we call them as purines? The purines is a heterocycle and heterocyclic structure. It contains two types of rings. One is a six-membered ring, other one is a five-numbered ring. One ring, the six-numbered ring, is called as a purine ring, sorry, pyrimidine ring, and the five-numbered ring is called as a imidazole ring. So it's a combination of two rings. That's why the term purine is given. Next, when coming to pyrimidines is made up of a single cycle. That means a single ring is present in their structure. So what type of ring? It is a pyrimidine ring, as I told you. So this ring is called as a pyrimidine ring. What are the examples for pyrimidines? The rest of the nitrogenous bases. So apart from adenine and guanine, whoever comes the other ones, who are those? Cytosine, thymine, uracil, all of them belongs to the pyrimidine structure. Okay, are you guys understanding? Okay, we have 13 people. Excellent. I didn't know this. Okay, guys, are you following? Yes, no? Can you confirm? Are you able to listen to me? Are you able to see the board, etc., etc.? Yeah. So let's go back here. So did you understand this? Next one is nucleosides. So what is happening here? When we are talking about the structure of the DNA, I told that ideally we have three important components that makes up the structure of the DNA. One is your nitrogenous, the deoxyribose sugar. Second one is your nitrogenous bases and a phosphate group. So we understood roughly what are these. So now we go for the assembly of this. Assembly means we have the component one, two, three. We have to assemble them to make one big structure. So that happens by beginning with the formation of a nucleoside. Okay. So first the formation of a nucleoside will happen. So what is a nucleoside? You take a pentose sugar. What is our pentose sugar? Deoxyribose. So I take my deoxyribose and I add my nitrogenous base. So deoxyribose plus a nitrogenous base is called as a nucleoside. You get my point? So where does this particular nitrogenous base occupy its position? As I told you, if you look at the structure of a pentose sugar, excuse me, okay, give me a second. Sorry, I'm sorry, students. Okay, if I look at the structure of a pentose sugar, right? So look here. I have five carbon atoms, right? So I can designate my carbon atoms as C1, C2, C3, C4, and then I have C5. Okay, this is my 5 carbon atoms. So now what will happen? Each carbon atom one place. Now what will happen? These compounds will come and occupy its position. Okay, so now you can see here, our nitrogenous base will occupy the C1 carbon atom. So if you have difficulty to remember, you remember that the first carbon atom, third carbon atom, and the fifth carbon atom are very, very crucial in the DNA. Okay, in DNA, these 3 carbon atoms are very, very important. Which one? 1, 3, 5, like odd numbers. Correct, right? So in the first carbon atom, what occupies the first carbon atom position? It is a nitrogenous base. So a nitrogenous base comes here at the first carbon atom and it creates, okay, a N-glycosidic linkage. What kind of a linkage? An N-glycosidic linkage. Please do not forget the name of the bonds that is produced. Okay. Oh my God. I know that you guys are very much aware because you study for the CBSE examinations, but still some of the students make a common mistake, confusing with bonds. So please have this small attention, okay? So the C1 carbon atom, a nitrogenous base comes and makes a linkage. So this linkage is called as N-glycosidic linkage. Okay. So once the formation of this linkage happens, okay, now this structure, this whole structure is called as a nucleoside. Got it? No. Now, what is a nucleoside? You can see if I have a nitrogen base that is adenine, guanine, cytosine, or thymine, when they combine with the deoxyribose sugar, what will be the chemical compound? What is the compound that results into? It results into deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine, etc. And you can see that the change in the structure. It was an individual sugar group. Now it has combined with a nitrogenous base, and this whole structure is called as a nucleoside. Correct? So next we go for the one monomer. See, that is a nucleotide. So if I ask you, what is our DNA made up of? What is our DNA made up of? Now, what do you tell? You told that it is a polymer. DNA is a polymer. Now, what is a polymer? Polymer is something that is made of monomers. Correct? No, it is made up of monomer. Correct, no, students? So, what are our monomers? See, if I have a chain, for example, if I have any chain, it should be made up of small links, no? One link should be attached together to form a chain. So what is our smallest link? So this is our smallest link. So this smallest link is called as a monomer. Okay. I know that it is very basic, but for some people it might be really difficult to understand. So I'm helping the other guys too. Okay. So now we have many monomers coming together to form a chain. So this whole chain is called as what? It is called as a polymer. Okay, our DNA is a polymer. What kind of a polymer we are? We are a heteropolymer. Okay, why hetero? Because we know that our nucleotides are of 4 different types. We have adenine, A, T, C, G. Correct? No, A, T, C, G are the 4 monomers. So what are our monomers of the DNA? Our monomers of the DNA is called as nucleotides, guys. Okay, so sorry, this chat bar is disturbing me. Okay, it is called as what? It is called as nucleotide. So what is a nucleotide? Now we know that we have 3 ingredients, correct? No, one is our nitrogenous base. Our nitrogenous base is occupied in the C1, carbon 1 position. Now I told you that there is another carbon atom. Which one? 1, 3, 5. No, 1 went for nitrogenous base. I'll write it as NB. 3 will keep it as pending. And the 5th position is occupied by the phosphate group. If you do not if you still cannot remember this, just know that you come across this term called as 5 prime and you come across a term called as 3 prime end, or it is called as 5 prime phosphate 3 prime OH. Agree, no? By now you would have come across if this lesson is taught in your class. If not taught in your class, put it in the chat box, or it is not taught. So then I can go in detail. I'm assuming that you know this lesson already. Okay. So now what happens, guys? Always you know this, the 5 prime end is a phosphate and 3 prime end is a OH end. So using this 5 prime end as a phosphate end, you can remember something. What is that? We have one more component left. What is our component? Our component is called as a phosphate group. So this phosphate group, where it will come? It will come and attach to the 5th carbon atom. Okay. So it will come and attach to the 5th carbon atom. So when it comes and attaches to the fifth carbon atom, it creates a bond. So what kind of a bond is created? It creates phosphoester bond. Okay, it is called as what? Phosphoester bond. Okay, or phosphoester linkage is created between which one? The C5 carbon atom and the phosphate group. So when this linkage happens, so now you have the first carbon atom occupied by a nitrogenous base, the fifth carbon atom occupied by a phosphate base, and then you have your deoxyribose sugar. So what happened here? Here you have a complete nucleotide. Okay, so in a complete nucleotide forms, now our each nucleotide can be read like this. For example, your deoxyadenosine here. You see, your deoxyadenosine becomes what? It becomes deoxyadenosine monophosphate, deoxygynosine monophosphate, deoxycytidine monophosphate, deoxythymidine monophosphate. You understand my point? Yeah. So this is what happens with the formation of a nucleotide. So these nucleotides. Now, one thing I will tell you, one small trick. So that when it comes to RNA, we'll talk about it. Let me not confuse. We'll go in a flow. Okay. Now we have nucleotides. I told you that each nucleotide, you can see here, guys. So this is one nucleotide. This is another nucleotide. This is another nucleotide. Now you can see that they have all been attached with each other. Correct? No. Now, if I ask you to write a structure of DNA, how do you write? Now, generally, some people will take the textual diagrams. Before you read your molecular biology, before you attend your biology classes, if I ask how would you do, you would just put it like this. Correct? No, you would just put it like this. Or this is DNA. Okay, generic structure of DNA. You just write it like this. Even without knowing that it is a DNA, we have seen this structure a million times. Being science students, correct? No. So now what happens? Now I take this small, very immature DNA structure, and we can understand what is a polynucleotide strand out of it. See, there are two lines going here. That means two chains are involved for the formation of a DNA molecule. Now we are talking about how each chain forms. For example, this chain. Okay, let's assume that we are talking about this chain or this chain, any one chain. So how are one nucleotide is assembling with another nucleotide to form one chain. So this is called as formation of a polynucleotide chain. Poly means many, nucleotide means nucleotides, and strand means chain. Okay, chain. Now what happens? Each of the nucleotide here, whenever one nucleotide is interacting with the other nucleotide, two important carbon atoms come into play. Now remember, I told you that 1, 3, 5 is very, very important. I told you 5 is for phosphate group, 1 is for nitrogenous bases, and 3 is still pending. Now 3 is having a free OH group. You can see here, that's what I was going to tell in the last slide, but I avoided it. You can see here there is a OH group in the sugar, pentose sugar. So this is in the which position? This is in the 3 prime. That means third carbon atom is giving the OH group. You understand my point? And then at the fifth carbon atom, what do you have? You have is a phosphate group. I told you. This is the fifth. See here, C5. So the fifth carbon atom has what? It has a phosphate group attached to it. Now what will happen? See, this is the nitrogenous base above. This is the nitrogenous base below and below and below. Okay. Now let us take two nitrogenous base. The phosphate, see, the OH group of this, the phosphate group of this will link with each other. So now you can see here. Okay. See the OH group. This was the OH before. Now this OH group and the phosphate. This is the 5 prime. No, this is the fifth carbon atom, right? See the fifth carbon atom. I mean, like the fifth carbon atom phosphate group and the third carbon atom OH group, they combine with each other. Okay, they form a linkage. What? How do they form the linkage? When a phosphate group come and interact with the OH group, which is in the third place, now dehydration happens. What is dehydration? Removal of water. The water molecule goes out. One H2O molecule will get removed. When one H2O molecule gets removed, a bond forms between a 5 prime phosphate group and a 3 prime OH group. So this linkage, okay, yeah, this linkage will create what? Phosphodiester linkage. Guys, this is the small confusion we all make. You can see here. This linkage is called, yeah, this linkage is called as what? That linkage is called as a phosphodiester linkage. Now, the question comes here. when during a polynucleotide chain, during the formation of a polynucleotide chain, a 5 prime phosphate group and a 3 prime OH group of the fifth carbon atom and the third carbon atom forms what kind of a bond? Now, you guys know that it is something related to phospho, but the problem is you come across phosphodiester linkage more common in your chemistry textbooks. That's why you tend to make this common mistake. So in a polynucleotide chain, you're right, it is phosphodiester bond. But if you see what is the linkage between the fifth carbon atom and the phosphate group, then what should be your answer? It is phosphoester linkage. There is no dye. There is no dye when it comes to the fifth carbon atom and the phosphate group. You understand my point? But when it comes to your polynucleotide chain, remember it is phosphodiester linkage. Okay, so now I told you you have one nucleotide and then you have another nucleotide, the OH group of the above nucleotide and the phosphate group of the below nucleotide. Now what they will do, they will create a phosphodiester bond. So how is the bond formation happens? Okay, hold on. Sir, at the C1 nitrogenous base will join, then the C3 what will join. Okay, I understood. I think I understood your question a little bit. At C1 a nitrogenous base will connect, and then you're asking what will join at the C3 carbon atom. I think that is your question. Okay, now if I misunderstand your question, you can correct me, please. Okay, see guys, that's what I told you. I told you to remember three carbon atoms, correct? No, so what is that? That is your now I'll make a justification for their question. I understand why they're getting confused. C1, C3, C5, correct? No, so C1 is nitrogenous base. You understood this point. And the C5 is phosphate group, correct? No, this is the phosphate group. Phosphate group. Now you can see here. This is the phosphate group. Now, why the OH group is important, I told you, is because you will forget during the formation of the polynucleotide chain, which carbon atoms molecules are interacting. Now, to form a polynucleotide chain means to form a DNA strand, I need many nucleotides. No, so this is one nucleotide, this is another nucleotide, this is another nucleotide. Now, how are they combining? Now, they are combining using these carbon atoms. So, the C3 as C3 does not link with OH. C3 already has OH. You understand my point? Now, you can see here, guys. You can see here there is a C3 already having a OH. Now, this OH group of the carbon, third carbon atom, will interact with the this carbon atom, fifth carbon atom. Now I will make you understand. See, C3 carbon atom OH group, C5 carbon atom phosphate group, they interact and they create what? They create this phosphodiester bond. Now, why did I mention three? Because to understand for the formation of the DNA molecule, you require two important positions in the sugar group, which is that one is a C3 carbon atom, other one is a C5 carbon atom. Correct? No, C3 carbon atoms OH group, C5 carbon atom phosphate group. They come together, they form a phosphodiester bond, and a polynucleotide chain or a polynucleotide strand formation will take place. So, in order to remember, I mentioned that you remember three important carbon atoms, which are those C1, C3, C5. C1 nitrogenous base, C5 phosphate group. What is C3? C3 has OH group, which interacts with the C5, okay, to create a phosphodiester linkage, which results in the formation of a polynucleotide strand. Did you understand, Andaman and Nicobar students? Yeah, any doubts here? Other guys, are you present? Why everybody is so quiet? Yeah, usually you say something, right, and it's missing today. Anyways, chalo, we'll go for the next one. So what do we have to study? So we understood how the polynucleotide chain forms, correct? No, and then today, next, what we have to understand is what's next, guys. We understood the structure of DNA. I mean, like basically, the DNA is made up of two strands, and we understand how the formation of one strand takes place, correct? No, so we understood this is one polynucleotide chain. This is another polynucleotide chain. So how is this formation took place? We understand, but the study and the structure of the DNA is a whole another story. Now, what happens? Do you know that uh, among Watson and Crick, do you think they are alive or Do you think they're dead? I know that this is a uh, very unusual question to be asking, but I'm asking you this question. Do you think Watson and Crick are alive? At least one among them is alive. Dead may dead maybe. Watson is really healthy and alive. Okay, he had come to India, that near Kolegal, for Dalai Lama. That is, Dalai Lama, the Tibetan monk. So he calls this spiritual gathering. He brings the top scientists and they speak. So here Watson had come and attended. And when I was studying in masters, when I was doing my masters, Watson was alive and well. Even I was like you, I thought that these guys must be dead, but they're alive and well. And it's so nice to see these scientists who make a big breakthrough alive, right? We are not usually used to it. I'm just saying, okay? So that's crazy. So, anyways, they're alive, guys. Not dead, maybe they're really alive. So, anyways, what difference it makes is somewhat curiosity, guys. They're like heroes. Okay, they should be alive. Anyways, so now what we have is these Watson and Crick. They made a very tremendous breakthrough in giving the structure of a DNA molecule. Okay, so to study the structure of DNA molecule, we have a whole different difficulties here. So the first thing is the Watson and Crick, they worked on already performed X-ray crystallography. So somebody else performed the X-ray crystallography, but the person who performed the X-ray crystallography was not able to, elaborate what is the structure of, the DNA. So that's why it ended up in Watson and Crick's hand. And ultimately, Watson and Crick worked together and they proposed the model of DNA. Can anybody tell me who proposed or who performed the X-ray crystallography studies of DNA? Now, meanwhile, while you answer, I will tell you what is X-ray crystallography. So usually, whenever we have to study some macromolecules, okay, such as proteins or such as DNA or RNA, if you want to study some structures, usually today we are studying more with proteins. Now, anyways, we already know the structure of DNA. We go with proteins, guys. Okay, we go with proteins. So now, yes, exactly. Rosalind Franklin, very good. Somebody thought you well. That's very important because we give credit to everybody who has worked for this, right? So, Andaman students has given us a brilliant answer. That is, Rosalind Franklin was a scientist who performed the X-ray crystallography. So, as I was saying, so to study any macromolecule, we cannot directly study under the microscope. So, we require some high precision, more detail, more smaller, something that we can see at the atomic level. So, we have to do that. So X-rays are something that we can use to study, the molecular alignment or something like that. How do we do this? We do the technique of crystallization. So this crystallization technique, using this, you understand what is crystallization, right? You crystallize macromolecules like proteins, and then you like DNA or RNA, and then you subject it to X-ray, and then the shadow falls on the X-ray film, and then you can understand based on the density of the molecules. For example, oxygen and nitrogen, you can make a difference between them by their atomic mass or how much light they absorb or emit, etc. Right? So by that you cast a shadow on the film, and through the shadow you can understand what molecules are present, what atoms are present, how they are arranged, or etc. But Rosalind Franklin, unfortunately, even though he had a breakthrough uh, in 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 getting the perfect X-ray crystallography of the DNA, but he was not able to, describe it properly. But it took an ingenious efforts of Watson and Crick to propose the model of DNA. So they also won the Nobel Prize in 1962. Okay, so they called this double helical structure, which is called as a helical DNA or a coil-like structure. So this is probably the most what do you tell, common thing that you have studied. Probably you might be very much aware of this particular topic, but anyways, I will try to be as brief as possible. Okay, we know that according to the Watson and Crick, they said that the DNA strand consists of two the DNA molecule consists of two strands. Okay, it is made up of what? Two strands. And these two strands, they are antipolar in nature. So antipolar in the nature means if one strand is having the 5' and 3' direction, other strand goes from 3' to 5' direction. That means what? Antipolarity, like anti-parallel, I can say. Okay, if the engine, the two engines are opposite, similar to that. So this 5 prime end and the 3 prime end comes with the free phosphate group and the OH group of your sugar. I just told no, in the 5 prime or the fifth carbon atom, there is a phosphate group. In the third carbon atom, there is a OH group. That is exactly what we're talking. If you look at the end of one DNA strand, so one DNA strand, they might have the fifth carbon phosphate group popping out. Okay, so if you look at the other strand, okay, if you look at the same direction, the other strand, you won't find the phosphate group. You will find the free OH group from the third carbon atom. So that's how we can say that it is anti-parallel in nature. Okay, so that is first thing. Second thing is, what is the composition of the DNA? So if you look at the DNA structure is a very, very beautiful molecular structure you can ever come across in entire biochemistry. So why? Because if you see here, the way it is arranged, in spite of having your nucleotides completely messed up. For example, you have purines, you have pyrimidines, some have two rings, some have one rings. How is it possible for the DNA to be evenly sized? Correct? No. So I will tell you what this statement means in a while. So if you look at here, just imagine a ladder. Okay, just imagine a ladder. Sorry, the lines are not straight because it's slippery. Okay, anyways, just imagine. Hold on. Uh, Excuse me. Yeah, just imagine a ladder like this. Okay, two ladder, like a ladder. And if you look at a ladder, you can see something like this. No, now just imagine the stick. This stick, let us call this as the backbone. Okay, let us call this as backbone. So this is our backbone now. Okay. Now, what is the DNA's backbone made up of? This DNA's backbone is made up of phosphate. It is made up of phosphate group and pentose sugar. That is your deoxyribose sugar. Okay. So these two things, that is a phosphate group and then your pentose sugar, is made up of, that makes up the DNA's backbone. Now here you can see here. Okay, here you can see properly. So this whole thing is made up of what? Our pentose sugar. So how is it even possible? Just look at this, guys. If you look at the, nucleotide chain, polynucleotide chain, you would be understanding. So this whole thing we are talking about, which one? This whole thing, this whole part. Okay, even though it is likely cross tilted, don't mind that. Okay, now you can see here, this is our sugar group, this is our phosphate group. Now what are they making up? They are making the stick. So this stick or this backbone is the DNA's backbone. So what is the DNA backbone made up of according to Watson and Crick? Watson and Crick says that the Watson, hold on, I have a text message. So, sir, why do we call that as 3 prime and 5 prime? What does it mean? See, it's a terminology. Some will tell, I don't know, I don't remember the term, but I have heard that, 5 dash end, 3 dash end. So that's what people used to call. Okay, I remember long back, even when we were in 12th. So I remember, one of my teacher referring to it as 5 dash end and 3 dash end. It is just to know the direction. Okay, so we call it as 5 prime end and 3 prime end, to make sure that it designates the direction. Okay, so that's it. It does not have a significant meaning, but we are talking just to bring a understanding of a direction. That's it. Okay. So apart from that, it does not have much significance, guys. Okay. So yeah, let's get back. So the DNA's backbone is made up of. So you understood what is a backbone, right? So backbone is something like a stick, like we have here. So this structure. So what is this structure made up of? This our structure of the DNA. The backbone of the DNA is made up of a pentose sugar and a phosphate group. Now we know that at the C1 carbon atom of our sugar group, there is a nitrogenous base. So now look here. Now look here. If you look here, you have your nitrogenous base. So this nitrogenous base will make bonds. What kind of bonds? These are hydrogen bonds. Okay. So these are hydrogen bonds. So they make these hydrogen bonds. Sorry, I have to keep changing the slides. See here. So these nitrogen bases, they create these hydrogen bonds like this. You understand my point? So that means what? They say that the backbone of the DNA is made up of a pentose sugar and the phosphate group, whereas the nitrogenous bases, they project towards inside. So now you understand what is projecting towards inside. So if this is the stick, if this is the pentose sugar, okay, if this is the pentose sugar, the nitrogenous bases are projecting towards the inside. So that is the meaning. That means the stepping sticks that you walk on the ladder is the projections of your nitrogenous base. The sides that are present are your pentose sugar and the phosphate group. You understand, right? Now, what about the nitrogenous bases? The nitrogenous bases, they exhibit the complementary nature. What is a complementation? Base complementation is a very important criteria of our DNA. What is base complementation? Always A makes a bond with T with two hydrogen bonds, and a cytosine makes three bonds with a guanine. And that means A goes always with T, complements, we say. A always complements with T and C always complements with G. So what is the meaning of complementation, if you ask? Or if you ask me what is the meaning of base complementation? For example, if I buy a chapati, I'm complemented with curry, right? I cannot eat a raw chapati. So always a chapati comes with sagu or a curry or a gravy, something like that. So that is it, that complementation. I go with you, you go with me. So A goes with T, C goes with G. So this property is called as base complementation. You understand my point? And they are beautifully made of hydrogen bonds. So A has two hydrogen bonds and CG has three hydrogen bonds. You understand my point? So these double and triple bonds are there. And remember, so these hydrogen bonds are a very crazy character. Why? Because you can see our DNA molecules are compressed, stretched, and they are wound very different ways during different phases of the cell cycle. But have you ever thought why this DNA strand is so rigid even during the cell division while condensing and decondensing, whatever you do, you don't see the cases of breaking in a, I mean, most of the organisms. You know that when it breaks, the whole cell get destroyed. But why not, if you ask the question? These hydrogen bonds are something that really helps the DNA. So they act like shock absorbers. That means when the DNA strand is compressed, these hydrogen bonds, instead of being straight, see like this, the hydrogen bonds will be there, no? So they can bend like this. So that gives the DNA extra stability as a structure. Okay, did you understand? So that is another thing. Now, we understood the base complementation and we see how the complementation works. So now I told you A always, so if the 5 prime end to 3 prime end, this is the base pairs, and 3 prime end to 5 prime end, these are the base pairs. You understand, if there is A, you always write T. If there is G, there is always C. So that is how. If they give you one strand like this and they ask you to write the other strand, please note the directions here. If the direction is given properly, and then you can choose the correct answer. This you will never forget. A, T, C, G, you will never forget. I know that. You never made a mistake. But you guys have already, I mean, like the past people have already always made the mistake while not observing the polarity. So they don't observe the 5 prime or the 3 prime. So that there they make a lot of mistakes. Okay. So now if I look at my DNA is like kind of twisted, it is turned. It is turned in a very specific direction. So this specific turn of my DNA is a right-handed helix. So if you look at it, it always goes from right to left. It always the turn starts from the right and it ends up at the left. So that's why we call it as DNA. Our structure of the Watson and Crick model is also called as right-handed helical DNA. We call it as what? Right-handed helical DNA. So what is this right-handed helical DNA? So that means my both strand turns from right to left. Okay. And it's like a staircase. It's arranged like a staircase. So let us understand how these staircases result in. Now, if you look at each turn, if I take the distance between each turn, so within this each turn, I end up 10 base pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So there are 10 nitrogenous bases on each strand. So there are 10 base pairs. When I say base pairs, pairs means what? You know that 10 and its pair, opposite pair. So I have 10 base pairs like this. Okay. Now, if you see here, the whole turn, the distance of the whole turn is what? It is around 34 degree angstroms. Okay. And each of them have 10 base pairs roughly. So the distance between each base pair, you can divide it. It is 0.4 degree angstroms. Now, my question is the information given on the slide according to the angstroms correct or wrong? Tell me. It should be nanometers or it should be angstroms. Now, if you tell me this, then I will tell you it's right or wrong. Now, is this right? Is this right or is it wrong? Did you guys understand my question? Let me wait for your answer. Please, guys, only 1 minute. Come on. Can you tell me? Is it 34 nanometers, or is it 0.34 or 3.4 nanometer? What is it? Can you tell me? Is it right or is it wrong? If I ask you to write in nanometers, what is the distance? What's the ideal distance? You didn't understand? No? Students, my question is, you have the length of the DNA, right? I mean, like one turn to another turn. Now it's given in angstroms. So now I'm asking, whatever the value is given in the angstroms, is it right? So if it is right, what is the value in nanometers? So that is my question. Did you understand this? Okay, come on, time is up. See, one more thing as you answer this, please make sure you come to the class on time, guys. We lost a lot of time, 0.34 nanometers. So 0.34 nanometers, what? So each turn, each distance between each base pair is 0.34 nanometer, and the whole turn is how much? 3.4 nanometer. Correct. So this is correct. Okay, so always yes, yes. So yes, 3.4 nanometers, that is 10 to 9. What is it? Yeah, 3.4 nanometers is the whole turn. 0.34 nanometers is the distance between each base pairs. Whereas if you look at the angstroms, in angstroms it is like this. That is 34-degree angstroms the whole length, and 3.4 degree angstroms is between each base pairs. Absolutely right. Okay. So if you know this, it's very, very important. So you know the values, right? So whenever it's coming in the question paper, you always observe this. All right. So let me not kill your time. I don't know what's coming up in the next schedule. We'll end the class today. And before you go, please wait. But sir, why is it given in angstroms? That's that. See, each textbook is different, guys. So the standard way of taking the distance is in angstroms, because of the considering the size of the DNA. We have to do that because it helps us in measurements. It help us in mass spectroscopy and many of the physical quantifications of the macromolecules. We need these angstroms. But unfortunately, in some text, in text and papers, they try to give in nanometers, considering your mathematical skills. So that's why we have to consider. But a standard way of expressing distance between each molecule is angstroms. Okay. Hold on. Before you leave, please do this. Okay. Guys, please attend the class on time. A lot 15-20 minutes is a huge loss, right? I can't finish my syllabus and I can't give you more info also. Okay. Thank you very much. Have a nice day. And after your voting, you can finish the class. So somebody gave me one. How easy was it to understand the concept? So please mention your problem. Okay. So I can understand really and help you out. So I'm here to help you out. So please make use of it. Otherwise, I'll be really, really hurt. Because I don't know what's the problem. Sir, there will be class tomorrow. Tomorrow, no biology. Other subjects will be there. Day after tomorrow is my class. Okay. So it's like that. Okay. Thank you, guys. Thank you very much. Yeah. The guys who are feeling difficult, please reach out to me. Okay. Feel free. It's not that we are in only one class. We have to, yeah. Thank you. Thank you very much. And yeah, it means a lot. Yeah, please reach out, guys. Yeah, that's very, very important for me, as a teacher. All right. Finally, it was nice to have you guys. Have a nice day. See you.